Okay, so pregnancy is going to be counted from the last normal menstrual period. This is because it's something that we can actually like measure, as it's something that is very clear and evident. And as such, because you won't be having the shedding of the endometrium when pregnant, this is why we do it from the last normal menstrual period. Now, what you will notice though is because of this very fact though, we're going to be adding another 2 weeks onto the pregnancy cycle. So from LNMP, it's going to be 40 weeks, but technically pregnancy is going to be 38 weeks long. So here's just some key days. There are more days to be learned than this that are covered in the lecture, but these are just the really key ones. So day 0 is when fertilization happens. Day 7 is implantation, and this is where the syncytiotrophoblasts secrete ACG. Remember, that's our LH-like hormone, which is going to be sent to the corpus luteum. Now in day 12, we're going to have implantation. This is going to essentially be completing around this time. We're going to have the primitive streak formed from days 13 to 14, and then gastrulation from 14 to 16. On day 17, we're going to have organogenesis, and then up to week 40, we're done. So a preterm baby actually will be fine. If they're born after 28 weeks, they can still survive. So essentially, those are just your big landmarks, and now let's go into detail. So before we do that, we're just going to talk about how the sperm actually penetrates the egg and fertilization happens. So ejaculated sperm can't immediately fertilize the oocyte. So what ends up happening is that when they enter the vaginal canal, certain hormones like estrogen can actually destabilize the sperm plasma membrane, and this essentially removes the protein coat that surrounds the sperm head. Now this is very important because what it does is it exposes binding sites, and that could be an answer for, let's say, 1.5 or 1 marks. Now something that is very important is the acrosomal reaction, and this is essentially how the different enzymes are released to break down the zona pellucida and corona radiata. Now, what ends up happening is that we have the swelling of the acrosomal layer, and the outer and inner layer fuse. This is what actually opens the pores for the enzymes to leak out. And like I said, it's easier, it's important for fertilization just so we can break through the corona radiata and zona pellucida, which essentially surround the egg. Now, there are 2 enzymes that you need to know. The first one is hydrolyronase, which essentially is going to break the outer corona radiata, and then acrosin, which is going to digest the inner zona pellucida. I used to always get confused between what's the outer and inner layer, and I thought of a really easy way to remember this. So I like to think of radius from the center of a circle all the way to the outside. Therefore, the corona radiata, which kind of sounds like radius, is going to be the outer layer, versus the zona pellucida is going to be the inner layer, because a zone, especially in basketball, is usually like a small area. At least that's how I like to think of it. So zona pellucida is the inner layer, corona radiata is the outer layer. So after one sperm digests the zona pellucida, we get a polyspermy block. So preventing multiple sperm from going in, we're blocking it, polyspermy block. And this is important because we don't want multiple fertilizations happening. We only want one sperm to merge with one egg, so we get the correct amount of DNA and a proper human child being formed. So we end up having 2 blocks. We have a slow block and we have a fast block. The fast block is going to be mediated by intracellular calcium increasing, and it's going to be an electrical block, and it's going to immobilize any nearby sperm. Now the slow block takes a lot of time to happen, hence we have the fast block. But the slow block includes hardening of the zona pellucida, which removes the binding receptors. Now let's start talking about how this cell is going to start dividing. So the sperm and the egg have fused together. You're going to get this morula stage, which is going to be 10 to 32 cells. You're going to get a blastocyst, which is 64 plus cells. Yes, you need to know those numbers. And then you're going to get this trophoblast, which is then going to turn into a cytotrophoblast and syncytiotrophoblast, which is going to be the actual cell that's invading the uterus. And this is going to be covering implantation. So this is going to be our syncytiotrophoblast invading the myometrium here, I guess, or the uterus really. And there are a couple of key things that we need to know. So this bottom part is called the yolk sac. I like to think of it as the bigger portion, therefore it is a sac, and it's kind of easier to remember it that way. You have this amniotic cavity, which is blue, I believe, in the lecture slides, in this tiny little area here. The cells right here under the amniotic cavity, or the ones that surround it, are called the epiblast cells, Epi is usually top, so on top of the amniotic cavity or most outside too. That makes sense. And then you have the hypoblast. And hypo doesn't mean under, but for me it makes sense to say under the epiblast cells, at least in this diagram. So those are going to be the hypoblast cells. You don't really need to know what each of these cells do in terms of histology, but you do need to recognize their overall functions. And essentially, the amniotic cavity is going to be protecting this the cell from physical trauma. It's going to be maintaining temperature. The yolk sac is going to be providing brief nutrient support. And then you also have your epiblast and hypoblast cells. Again, refer to lecture for key specific functions. Now, later on, we're going to have this allantois form from our yolk sac. And all you need to know is that this basically forms the umbilical cord later on. Kind of makes sense because the yolk sac is what is providing that nutrient support. And again, this is a very big summary. I think this was like 4 or 5 slides. So definitely pay attention to the details there. The primitive streak is going to form. And essentially the primitive streak is, it's not the wrapping of the yolk sac around the amniotic cavity, although it does look like that in the image. All we're doing is we're taking a cross-sectional view like this. So imagine your eyes are right here and they're taking a cross-section from the top down. And because the amniotic cavity is physically a little smaller than the yolk sac, that's why in the picture it looks like this. So this is going to be your amniotic cavity and the yolk sac on the outside. It's just a top-down view. So essentially, what is the purpose of having this primitive streak in the amniotic cavity or surrounded by the yolk sac? All this is doing is establishing a direction. So it allows us to know what's the head and what's the tail of the embryo. And essentially, then we're going to have gastrulation. Gastrulation is just producing these different germ layers, and I believe MSS goes into a little more detail about this. Now, in terms of repro, there are 3 different germ layers that you need to know. The ectoderm is going to produce the skin and nervous tissue. The mesoderm is going to produce muscle, so M and M. It's also going to produce bone. Kind of makes sense. The muscles attach onto the bones, and most blood vessels, I guess, because they go through the muscles. Now, in terms of the endoderm, you're going to have epithelial linings. The respiratory systems and urogenital systems are also going to come from the endoderm. Endoderm is basically everything other than the ectoderm and mesoderm as a very broad generalization. And yes, you do need to know at least 3 examples for each one of these.